Hello friends, good morning. In the previous lecture, we discussed about microtechnique, which are the basic steps in microtechnique. So which are the steps? The first step is collection of material. After collection of material, the animal can be killed. Killing means sudden and permanent stoppage of life processes. त्याला आपण काय केलं? किल करून टाकलं. Life processes are completely stopped. Then after collection, the tissue can be fixed into the fixative. Hence the second step is called as fixation. The third step is called as washing, that is removal of fixative from the tissue is called as washing. Then fourth step is called as dehydration, that is removal of water from the tissue is called as dehydration. Generally, it can be done with the help of alcohol. We are using upgrade series of alcohol, that is 30% alcohol, 50%, 70%, 90%. Then tissue are transferred into the absolute alcohol. By keeping into the absolute alcohol, the water is completely removed from the tissue. Hence, it is called as dehydration. After dehydration, we must have to remove the alcohol from the tissue. The removal of alcohol from the tissue is called as dealcoholization. For this purpose, we are using xylene. In xylene, the tissue becomes clearly transparent. Hence, this step is called as clearing step. After clearing, the tissue are transferred for infiltration. There are two steps of infiltration, that is cold infiltration and hot infiltration. Cold infiltration can be done at room temperature, while hot infiltration are done into the oven by putting the paraffin wax into the oven. Right? This infiltration is also called as impregnation. The second word for infiltration is also called as impregnation. After impregnation or infiltration, the tissue are embedded. Hence, the next step is called as embedding. By embedding, we can able to prepare the block, tissue block. And when the tissue block can be cut into the rectangular size, then they are called as trimming. This process is called as trimming. Then after trimming, the tissue block are attached to the microtome peg. On the microtome peg, we are able to take sections. So we will get serial sections. This process is called as section cutting. After section cutting, the tissue are affixed to the microscopic slide. The microscopic slide ला affixing करणे म्हणतात त्याला. Right? After affixing, the slide can be kept for overnight. Then on the next day, the tissue can be or slide can be stained. This process is called as staining. Generally, we are using two types of stains, that is hematoxylin and eosin. Hematoxylin is called as nuclear stain, while eosin is called as cytoplasmic stain. This process is also called as double staining method, so that we will get a clear picture of tissue. Cytoplasm as well as nucleoplasm is seen under the microscope. Hence, this process is called as staining. After staining, the slide can be mounted with the DPX. DPX त्याच्यावरती एक ड्रॉप टाकायचा and put the cover slip on it. This is called as mounting. By this way, we are able to prepare the permanent slide. This is called as microtechnique. This microtechnique, what is the use of skill of microtechnique? Or they are called as applications of microtechnique. Applications of microtechnique. So you will get the skill. Which skill you will get? You are able to prepare the permanent histological slide. By studying this microtechnique, you will get the skill about how to prepare the permanent histological slide. ही स्लाइड कशी तयार करायची याचा स्किल तुम्हाला येतं. You are able to prepare the slides. Okay, then you will get the knowledge by studying microtechnique. You will get the knowledge about how to prepare the permanent histological slide. After preparing the histological slide, you are again study the histological structure of the slide. You will get the knowledge of histology of each organ. तुम्हाला प्रत्येक ऑर्गनची हिस्टोलॉजी पण मिळते. Thus you will get knowledge also. Then what are the job opportunities after studying the microtechnique? Hence the third point is job opportunity. So friends, you will get a job in health centers, that is public health centers or private health center, that is government hospitals, private hospitals. In that hospitals you are able to prepare the histological slides. You will get an opportunity in government as well as pathological centers also, pathological laboratories, civil hospital, government hospitals, private hospitals, where the microtechnique person can get an opportunity. Okay, so these are the applications of microtechnique. Then as far as our syllabus is concerned, the first chapter is collection of material. How this animal can be collected. The basic principle in the microtechnique is you should have to bring live animal into the laboratory. If you have to study rat tissue, then you should have to bring live rat into the laboratory. If you have to study frog tissue, you have to bring live frog into the laboratory. After bringing this live animal, it must be killed. The killing means sudden and permanent stoppage of life processes. So here, collection of material. So after collection, the animals can be killed, right? Killing and fixation are two different processes. Fixation means preservation of tissue and killing means sudden and permanent stoppage of life processes. Right? For that purpose, for killing we are adopting two steps, that is decapitation. There are different methods of killing. The first method is decapitation means what? That is removal of head or cutting of head is called as decapitation, or blowing of needle into the head. म्हणजे काय, हेड मध्ये हेड आणि मान असतं ना, नेक, त्याच्यामध्ये काय करतात, पाठीमागच्या बाजूने निडल इन्सर्ट करायची. दिस इज कॉल्ड एज ब्लोइंग ऑफ निडल इनटू द हेड इज कॉल्ड एज decapitation, or directly cut the head is also called as decapitation. After this what happens, the animals get killed. तो animal मरून पडतो, त्याला म्हणतात decapitation. After this decapitation, generally marine animals are dissected into the marine water. For example, if you brought marine fish and you have to study the histological structure of that fish, then this animal after killing they can be dissected into the marine water. If you are observing freshwater fish, then these freshwater fish are dissected into the fresh water, right? So this process is called as killing. For that purpose we are adopting decapitation method. The second method of collection, that is anesthesia. Actually, what is my anesthesia? Anesthesia means loss of feeling. म्हणजे त्याचं जे कॉन्शियसनेस आहे ते फिलिंग काय होतं, लॉस होतं इन ऑल पार्ट्स ऑफ द बॉडी or some part of the body. That is, doctors, they are giving local anesthesia. If we have to operate here in the hand, they are giving local anesthesia, right? Anesthesia means what? Loss of feeling in all or part of the body. For laboratory animal, we are giving chloroform. For anesthesia, we are using chloroform vapors, and by giving chloroform vapors, the animal its feeling is lost. Only respiratory and circulatory system are functioning. Other systems get stops. That is, its movement gets stops. its movement halchal band houn jail, and so that we are able to dissect the animal. If suppose you are dissecting the frog, you should have to dissect into the fresh water or running tap water. त्याला रनिंग टॅप वॉटर मध्ये डिसेक्ट करा, राईट? इफ यू आर ऑब्झर्विंग मरीन एनिमल, तुम्ही मरीन एनिमलचा अभ्यास करत असाल, दिस मरीन एनिमल कॅन बी डिसेक्टेड इन द मरीन वॉटर. दिस इज कॉल्ड एज एनेस्थेशिया. जनरली फॉर फ्यू एनिमल्स physiological saline solution is also used for dissection purpose. Marine invertebrates are dissected into the marine water, and the tissue are immediately removed from the animal. Those tissue which we have to study, तुम्हाला ज्या टिश्यूचा अभ्यास करायचा आहे, फॉर एक्झाम्पल इफ यू हॅव टू स्टडी लिव्हर, देन रिमूव्ह द लिव्हर फ्रॉम दॅट ऑर्गॅनिझम. इफ यू हॅव टू स्टडी स्टमक, देन रिमूव्ह द स्टमक फ्रॉम दिस एनिमल. It should be done immediately. Hast should not be done. Hast means what? घाई करायची नाही. The hast treatment make the tissue damage. घाईमध्ये टिश्यू काय होऊ शकतात? डॅमेज होऊ शकतात. You should have to practice about them. त्याची तुमची तयारी असली पाहिजे. मग easily you should remove the tissue, and immediately after removing the tissue are transferred to the fixative. म्हणून पुढची स्टेप काय? आफ्टर कलेक्शन, फिक्सेशन, राइट? बट सम invertebrate animals, they are contractile one, that is their body get contract and relax. Such animals are not mounted directly on the slide. For that purpose, they should have to narcotized. Hence, the third step is called as narcotization. Actually, what is my narcotization? Narcotization means some marine worms or freshwater worms, they are contractile one. They have ability of contraction and relaxation. So during this condition, these animals can be narcotized so that they are fully expanded. ते पूर्णपणे काय होतात? Expand. They get relaxed. Their smooth muscles get relaxed, and they are fully expanded. This fully expanded animals is called as narcotized animal. So these animals can be put into the magnesium sulfate solution. त्यांना कशात टाकायचं? मॅग्नेशियम सल्फेट सोल्युशन मध्ये, so that these animals get relaxed, and after fully relaxed you should have to able to mount them on the slide. त्याला तुम्ही स्लाइड वरती माउंट करू शकता. This process is called as narcotization, right? Here some of the animals cannot be dissected. Some animals cannot be dissected so that they can be directly mounted on the slide. त्यांना जसं तसं माउंट करू शकतो आपण. This process is called as whole mount preparation. त्याला काय म्हणतात? Whole mount preparation. Right? For example, if we have to prepare the slide of bedbug, slide of amoeba, slide of paramecium, euglena, these animals cannot be dissected. त्याचं आपण डिसेक्शन करू शकत नाही. सो दॅट these animals are directly mounted on the slide. त्यांना जसं तसं स्लाइड वरती आपण माउंट करू शकतो. Hence this process is called as hold mount preparation. We are also prepare the slide of mouth parts of housefly, mouth parts of mosquito. Right? मग असे जे माउथ पार्ट्स आहेत त्यांना डायरेक्ट माउंट करू शकतो. This process is called as hold mount preparation. Generally, these hold mounts can be prepared by how it can be prepared, where the animals are not directly mounted. First of all, these animals are collected. म्हणून त्याची पण अशी स्टेप्स आहे. collection of material. After collection, these animals can be what we call as fixation. Tissue or animals are fixed. After fixation, they also have to wash. That is washing. Then after washing, you should have to dehydrate them. That is removal of water is called as dehydration. That is removal of water from these animals. After dehydration, these animals are cleared. This step is called as clearing, or it is also called as de-alcoholization. Removal of alcohol from the tissue, or removal of alcohol from this animal. That is, they are called as clearing. And after clearing, these animals are directly mounted on the slide. The direct slide वरती काय करू शकतो आपण माउंट करू शकतो. Hence, this process is called as hold mount preparation. Okay.